Welcome to Brain Ponderings, conversations with scientists at the forefront of brain research. Today, it's my pleasure to welcome Dr. Erika Augustine. She's an expert on rare neurological disorders. Started out probably, I guess, more interested in adults and Parkinson's and movement disorders, but she's been focusing a lot on rare childhood neurological disorders, which are really kind of devastating because obviously they're affecting children, their families, and so on. And even though kids are much more resilient than us and seem to be able to take their problems better than I do, or maybe many of the listeners do, it's not good. And for most of these disorders that she'll talk about, there's no treatments. Although the one she'll focus on, there is recently a treatment. So welcome, Erika.
Thanks so much. And, I have to say, I completely agree with you. Children are resilient. They are just such a joy to be a part of the care for a child when there's a need. It's very different than taking care of adults who are also wonderful, but there's something special about children.
I have an experience so we moved, my wife and our kids moved to Baltimore area in 2000. And I was at University of Kentucky then. And one of my sons, actually his best friend, they went to elementary school together and they were just transitioning to middle school. And it just happened, his family moved to Bethesda at the same time, and their son got hit by a car and had a traumatic brain injury. He was in a coma, and he was at Kennedy Krieger, where you're at, which if people haven't heard of it, maybe you can tell them about it, but it's nationally and internationally known as like one of the best places for children that have brain, well, not just brain, but spinal cord, neurological problems in general.
Kennedy Krieger is a really unique place. We focus exclusively on care and support for people and families who are impacted by neurological and neurodevelopmental conditions. And there aren't many places in the nation or even around the world that have that very exclusive kind of focus. It drives everyone in the same direction, bringing their unique expertise, but towards that same population. And we do that through care, research, and education. Care in the way that you think of, outpatient and both inpatient services as well, very specialized inpatient services. Also through our research programs that bridge the bench all the way to bedside. And then education in ways that you might think of for an academic medical center, training residents and fellows, training folks, clinical disciplines of all kinds, and research education as well. But we also provide K-12 education for individuals with neurodevelopmental needs. And so we actually have schools that are an extension of the Baltimore public school system, so that those who have very specialized learning needs have an environment where there's expertise and their learning needs can be really well served.
So you were recruited to Kennedy Krieger. Your official title, at my understanding, is Associate Chief Science Officer and Director of the Clinical Trials Unit. Well, Director of the Clinical Trials Unit is a big deal. And later on, maybe you can talk about some of the clinical trials you've been involved in. But let's go over your background a little bit. So you're an MD, but I look back at like your CV, and your bachelor's degree was in anthropology.
That's right. I knew that I wanted to pursue a medical career. So I was pre-med going in.
Yeah.
There wasn't a pre-med major at Harvard College. And so I wanted to both fulfill all of my pre-med requirements, but also have the opportunity to explore other kinds of knowledge. And so biological anthropology allowed me to do both. I got a chance to think about evolution and the development of cultures and societies and what it meant to have community through a biological science lens. But at the same time, yes, I covered my pre-med sort of needs, but it really did allow me to explore the school in a way that I think it would be more difficult or more challenging otherwise. And I still have a lot of those interests. I think as my career has gone on, I have some ethics interest, and there are these other sorts of disciplines and types of expertise that really that really capture my interest.
And then you went to medical school at University of Rochester, and you were interested at that time when you went to medical school in pediatric neurology, or was that something that developed later?
It developed later. I again knew I enjoy children. I enjoy caring for children, and thought that I would pursue a pediatrics career, a pediatrics focused career or subspecialty. I think pretty quickly I realized I was interested in a subspecialty. I have this immense respect for generalists. So internal medicine physicians, pediatrics physicians, med peds folks, they have to have such an incredible breadth of knowledge about everything. Being able to manage the day-to-day and then also be able to immediately recognize something that is serious and needs more evaluation. And I wanted to be able to have more focus and was looking for a subspecialty. My approach to those third year rotations was, if there's a pediatric version, that's where I want to be. So pediatric surgery, pediatric psychiatry, pediatric neurology, which I didn't actually even know was a specialty until I got to that year. I'd really enjoyed our mind-brain behavior course. It's phenomenally taught by Ralph Josefowitz at the University of Rochester and was for a very long time. But child neurology, that rotation opened me up to the field, which is very different than adult neurology. And I really found everything I was looking for in a medical career during that rotation.
Yeah. Okay. And then when did you first get involved in research?
That was during my clinical fellowship.
Okay.
So I got to the end of my residency training, and I was in that last year really thinking about, how do I want to spend my time? What's the patient population, I'd like to focus on? And I wanted to learn more about movement disorders. And I thought about, what are the places that I could go? And I decided to go back home for several reasons. I understand you were also from Rochester, but Rochester, Minnesota, not
That's
Rochester,
right.
New York.
I went to Mayo High School.
Well, I went to a high school outside of Rochester, so I've been in and around the University of Rochester for a long time. And Jonathan Mink was there and was leading the child neurology division, and was and is one of the world leaders and expertise in movement disorders. And so it was just a logical next step to go to a place where I had a home base, where I had roots, and I had a faculty member who would invest in me in terms of supporting my career and teaching me. And so I went for clinical training, and I think I must have been in the interview and someone asked, well, what else are you interested in? And I said, well, I've always wanted to learn more about clinical trials. I don't really understand how they come about, who develops them and designs them, how one gets involved, how are drugs made? And they said, oh, we do that here. And so you should go talk with Birch Griggs. And for, at this point, it's more than 30 years. He has run a T32 NIH-funded experimental neurotherapeutics fellowship. And so I had the opportunity to apply and then combine clinical fellowship with research training. And that's really how I got started. It's also part of why I have some Parkinson's work that I did. It was part of that fellowship. But it was just a really phenomenal environment to train clinically and to start to build a research career. It was wonderful.
Yeah, that's great. And then only recently you moved to Baltimore, right?
That's right. I moved in 2020. It was an interesting time to make a move in the middle of the pandemic that was happening.
Well, maybe it was a good time, actually.
I agree. I think you could see it
Because
both ways.
all their clinical trials were shut down, right?
Yeah, that's right. And so things were pretty slow. And so it gave me time to get to know the Institute, the people, really develop a vision for what we'd like to do and where we want to go. And I think gave opportunity to start to see the needs without the onslaught of there being so much and so many changes happening in clinical trials. We could build a little bit of a foundation before things ramp back up again. And they started to get busy again, I would say 2022 kind of timeline. And so now we're full steam ahead, which is great.
Okay. Rare neurological disorders. There are disorders, and most of them are genetic. Is that true or not?
Yes. And, we see that interest for me in rare diseases came about through my movement disorders interest. So pediatric movement disorders in particular, a lot of your patients will have tics and Tourette syndrome. A lot of people will have tremor. And a number will have perhaps not common, but neurodevelopmental kinds of movement disorders that may come at particular periods of childhood that aren't necessarily a problem, but can be striking in appearance. And everyone else has something rare. And so that's where that interest was sparked and how I started to think about rare diseases. And I was really compelled by the fact that so many of the patients that I had, I needed to do a comprehensive genetic and metabolic evaluation. Each had something unique. And then the tools that I had for treatment were fairly limited. So rare diseases encompass thousands of conditions, somewhere in the realm of maybe 7,000 to 10,000 different, unique, distinct diseases. The majority are genetic. The majority start in childhood, and the majority have some sort of neurological symptoms. So I sometimes say rare diseases are the diseases of the child neurologist because so many folks come to us for care.
Yeah, I was a lab chief at NIA for a long time. And one of my colleagues there, Will Bohr, he did a lot of work on the premature aging syndromes like Werner and Bloom, and they have very major, in some cases, neurological aspects to it. Okay, so about 7,000 total. What about neurological? You're saying most have some neurological, but are there, I guess, what are some of the most prominent, I guess, brain-centric rare disorders?
We can think about those in groups. Examples, there are some of the neurocutaneous syndromes, neurofibromatosis, tuberous sclerosis, some of the neurodevelopmental syndromes like Fragile X. That might be one of the most prevalent genetic etiologies of neurodevelopmental diseases.
And that's considered to be like on the autism spectrum, right? The Fragile X?
There can be this overlap. So there are a portion of individuals with Fragile X syndrome who do have a frank traditional autism spectrum disorder phenotype. Rett syndrome is another one where, again, autism spectrum disorder can be a core set of symptoms, not necessarily for everyone, but for some. And then I think because there can be similar impairments to autism in communication, in restrictive interests, in movement disorder, or even repetitive behaviors, many neurogenetic syndromes, when they start early in childhood, autism might be the first diagnosis. And so I think now as a standard, when a child has a new diagnosis of autism, those providers really need to think about what is the genetic workup that needs to occur for that person, because the diagnosis doesn't stop at autism. That may describe the clinical syndrome, but there's still a need to look for an etiologic or biological cause of those symptoms, and those can be quite varied. I think it's in some ways the definition is akin to how we might think about Alzheimer's disease, although that's evolving in sort of a biomarker era. But starting out as a set of clinically defined syndromes, many psychiatric syndromes based on clinical diagnoses of a certain set of syndromes. And I think across neurology, across aging syndromes, we're now moving towards biologically defined diagnoses.
Yeah, and then that's very important when you consider how to develop treatments too, right? Because you can have multiple well, multiple genes that lead to autism, like, social withdrawal, repetitive behaviors. But as far as treatment, they're different, since they're different pathways may lead to the same kind of phenotype. It's important to know which gene's affected and how that abnormal gene, the gene mutation, leads to the disease, because it may lead to disease-specific treatments or a treatment that will be effective in a subset of kids that have those symptoms and not in another. So is genetic screening becoming is that really common now?
It is. So for all of these sort of, again, more clinically defined, you have this many symptoms of this type, this many symptoms of this type, diagnoses that are made in that way more and more. Once that clinical diagnosis is made, the genetically defined or the biologically defined diagnosis is then sought. That's true for autism spectrum disorder. It's true for other more broad intellectual and developmental disabilities. It's true for epilepsy, and then also true in the case of movement disorders, or, again, all of these things that we diagnose based on what we see. That next step is often genetic diagnosis or an attempt at genetic diagnosis. Cerebral palsy is another example of that.
Okay, Batten diseases. What
Mm-hmm.
are they? What's known about what's going wrong in brain cells?
Well, these are a sort of a perfect example of conditions that we once thought were just one, and we now realize are many. These are a group of lysosomal storage diseases that are known for primary central nervous system dysfunction, not exclusively, but they're primarily CNS diseases. And they manifest with a variety of symptoms, but there are some core common shared features of vision loss, epilepsy, movement disorders, and dementia. And across the group of conditions, to varying degrees, those core 4 features are shared. They tend to start in childhood. They tend to result in shortened lifespan, and pathologically, they are characterized by lysosomal dysfunction, by lysosomal accumulation of lipopigments. And that's really what unifies this group of conditions. It's those clinical features combined with that storage of lipopigment intracellularly that brings them together as a specific set of conditions. There are at least 13 different NCLs, and that list seems to grow periodically.
I've had multiple podcasts that have touched on, well, some focused on and some touched on autophagy and how cells normally get rid of like a damaged membranes or lipid components or proteins or even organelles. And so what are the genes and how are they're affecting the lysosomes? Are they affecting the pH? So the lysosome's like an acid bath in the cell where things are shuttled to degrade them. Is that what's happening, or is it something else?
It really varies. So I mentioned there being perhaps 13 different NCLs. Each NCL is defined by a unique genetic etiology, and those individual genes encode proteins that may have very different functions or even maybe different types of proteins. Some of them are secreted enzyme. Some of them are transmembrane proteins. Some have traditional, you sort of go back to what we thought of as the sole function of the lysosome long ago, really being about protein degradation, protein recycling. So some, like the CLN2 gene, that encodes tripeptidyl peptidase, that enzyme has sort of a very traditional lysosomal function. It cleaves proteins into peptides. And so that in that way that we think about the lysosome and its function, some of the NCL genes fit very classically with that. But in our more expanded view of lysosomal function in terms of regulation of proteins and that regulate lysosomal pH, or that may play a role in immunity through roles in antigen presentation, or roles that have to do with intracellular targeting and trafficking. There really are these very there are a variety of functions, including nutrient sensing, that we now know that the lysosome plays a role in these cellular functions and maintenance of cellular homeostasis. And the NCL proteins, some of which are very well known exactly what they do, they do actually have functions that distribute across those various lysosomal functions. And then there are some NCL proteins like the CLN3 gene that encodes Batten. We're a little less clear on exactly what Batten's function is. There are some hypothesized mechanisms, but for some it's a little bit less definite. But I think it's interesting that there's not, even with that common phenotype of vision, epilepsy, movement, and dementia, the proteins themselves that where deficiencies result in these diseases, they have very different functions.
So you mentioned nutrient sensing. My lab over decades, we did a lot of work studying the effects of intermittent fasting on the brain. And one way to, actually it's the way it was discovered, autophagy was discovered, is to remove nutrients from cells. Initially in culture, that got a Nobel Prize actually, but if you have animals fast and then you look at autophagy in brain cells, it's upregulated. And I'm wondering, so kids, normally you wouldn't think of like restricting the energy intake of a developing kid. But I'm wondering if there would be if this might be some way that knowledge that, so about the mTOR pathway, right? And it's kind of antagonistic to autophagy, and the mTOR pathway is activated when you're fed, in the fed state, when you got a lot of nutrients in your blood. Then in the fasted state, mTOR goes down, autophagy goes up. Has people in the field thought of that or shied away from it, or could you get it through a internal review board, or is there enough consistency between individuals that you can actually do clinical trials and see if an intervention is slowing the progression? So I know I'm asking a lot of questions, but
so I'll start at the end on the clinical trials end. So there have been clinical trials in the NCLs, and over the years of varied targets and varied designs. And it's really more recently that we've gotten into more externally controlled, larger scale prospective studies. A lot of retrospective, a lot of case series, a lot of open label, but we do see the science, so to speak, of clinical trials advance in this arena. In the 70s and 80s, knowing that there were potential deficits in lipoproteins, in fluid membranes, that there was a lot of interest in antioxidants. There was a lot of interest in fatty acid supplementation. And so those were some of the early trials. It's interesting that you bring back, I don't want to say a dietary intervention necessarily, but dietary modification as a means to potentially impact disease. It's something that we actually hear a lot from families, a lot of questions about intermittent fasting, questions about the ketogenic diet, both as a role in epilepsy or in disease modification. And it's something that we have not yet advanced to really being able to well target those questions in this specific population. I think the field as a whole, the readiness of the community for clinical trials is at a different state. And this may be the time where we can actually start to look at those more nuanced interventions, even on the individual level.
Yeah, and since they're rare, it's not like Alzheimer's or Parkinson's where it's relatively easy to get enough individuals to have a big enough N and you can do statistics and
Right.
so on. So it makes it a lot harder. So in that
These are
regard,
very small studies. I mean, so a large study in this setting might be 20, 30, 40 people.
yeah.
So that's a big Batten disease study. And so trying to account for the intra-individual variability is a real challenge
Yeah.
in thinking about that, especially as it relates to a comparison group. How well matched can they be, even if they're matched on a host of factors? Are they really an appropriate or adequate control? And then when that control is historical, as it has been for some studies, again, how do you make sure that's a well-matched group and is it appropriate? Has been difficult.
Yeah, and since there's inter-individual variability, really you have to be able to follow them out for a long time, I would think. And, with but having said that, so there is a recently, yeah, I guess FDA approved treatment for Batten. Can you talk about that?
Yes. So in 2017, the first disease-modifying treatment for any Batten disease was approved. And it was approved for CLN2 disease, the form that encodes tripeptidyl peptidase that I mentioned, so soluble enzyme. And the idea behind the therapy was to replace the enzyme that is deficient or missing altogether. And that approach, replacing deficient enzyme, is one that has been long used in lysosomal storage diseases like Fabry disease, Pompe disease, Gaucher disease, mucopolysaccharidosis. There's a long history of enzyme replacement therapy, but the central nervous system had always been a challenge.
Yeah.
Systemically administered or IV, intravenously administered enzyme is too large to actually cross the blood-brain barrier. And so you didn't have the opportunity to treat the CNS, even in those conditions that I mentioned where there are CNS manifestations, you couldn't treat that protected space. And so this was actually a CNS targeted enzyme replacement. And so the route of administration had to be a bit different. So these are administered into the ventricles, placement of a Maya reservoir, as you might use for chemotherapy or for other relatively short-term treatments to allow for infusion of enzyme into the CSF and then treatment via intracellular or via cellular uptake by the mannose-6-phosphate receptor mediated endocytosis pathway so that cells could uptake enzyme, bathing the brain via CSF. And it has been quite effective. It's essentially been transformative for this disease, where in the classic onset, usually starting between the ages of 2 and 4 years of age, very rapidly progressing over a couple of years, and then an extended period in a severely impacted state with death that typically occurred in the second decade of life. Now, children in the second decade of life are walking, they are interacting with their environment. There's still a number of medical needs, but it's been a very impactful therapy. It also is a challenging therapy to administer. So families come in, the children who receive this therapy and some adults who receive this therapy have infusions that last several hours every 2 weeks at a center that's able to have CNS infusions and administration. And so it's not necessarily something that can happen everywhere. And for some families, in addition to needing to be at an infusion center every 2 weeks, they actually have to travel a fair distance sometimes to actually get to those centers. So there's a real, it's a major commitment, and it's not without its burden and also not without complications. So it's been a major advance, but there still needs to be more. you talked about targeted therapy and really being able to leverage a specific etiologic diagnosis for targeted treatment, and that's exactly what this is. So while this is one of the NCLs, it is only for CLN2 disease and is not actually a therapy for the other 12 forms of NCL because they do not have that same enzyme deficiency.
Okay. Now, ever since the human genome was sequenced, that as you probably know, when that happened, well, I'm gonna hold you, maybe not old enough, but but there was a lot of, I guess, maybe hype, but I guess promise of being able to cure diseases by identifying the genetic defect and correcting it. And there's been a lot of advances in how to do that. And a lot of this work actually starts in cell culture, then in mice, and moves on. There have been a few clinical trials of gene therapies for some disorders with some success.
Mm-hmm.
What's going on with these rare disorders? Because in theory, that would be, oh, now I guess since they're so rare, you can't do this. And they're not, I guess, they're not dominantly inherited. I mean, that's obvious, right? Because if they're dominantly inherited, they'd be more common. So these are recessive usually?
They're usually recessive. So there are some that even within the NCLs, there is one dominantly inherited form, but they are, for the NCLs, they are almost exclusively recessive. And for most childhood onset diseases, they tend to be recessively inherited disorders. There are exceptions to that.
Does that make it harder? I guess that makes it, wait a minute. So if they're recessive, does that mean that say the gene for CLN3, right?
Mm-hmm.
So you could have multiple mutations in that gene that all lead to the same problem, right? That gene dysfunction. And so then you would, do you think you would have to correct one allele, one mutation that was, say, from the father, and then they'd still be good with the mutation from the mother? Or it's a little more complicated from dominantly inherited, where you just have to correct one gene.
So I think it depends on the disease, and it may depend on the specific variants as well. And then there's the need for experience. So I would say in recessive conditions where the gene in question creates an enzyme, we measure enzyme level. And there we have some sense that, okay, individuals who are affected by the disease, they may have enzymatic levels or enzymatic function that falls in this range. And then we can see, well, heterozygotes who we don't think have any symptoms or don't have any substantial symptoms fall in this range. And then individuals who are not carriers fall in this range. And so you have at least a little bit of a guide or template, all other things being equal, that if you could boost enzyme at least into the heterozygote range, you should have benefit. Now, there are a lot of caveats with that as well. So is the enzyme where it needs to be cellularly, regionally? Is it temporally as well? So it's making a lot of assumptions. Can you actually have sufficient rescue? If you have crossed a certain point, have you had such neuronal loss, or if there are loss of other relevant cells that you cannot recover that back? So there are a lot of questions that remain, but at least for those kinds of conditions, you have a theoretical concept. I think when it comes to whether it's the NCLs or other recessive disorders, whether or not you actually need to correct both alleles or not it really depends on what the variants are. If you have two severe variants, it may not be sufficient to correct one. And it also, again, may depend on time, how much functional protein is actually required by the system in question. So that really comes down to the preclinical studies that help us better understand that, and then the clinical trials. However, if you are truly pursuing precision therapies at the individual level, targeting individual variants, and it becomes a real challenge to think about what is required, what preclinical evidence suggests enough promise that you should proceed into human study, and then there are other kinds of measurement issues in the human clinical trials themselves.
Yeah. You mentioned the ethical issues. One thing I thought a lot about, so I did a lot of work on Alzheimer's disease, and in that case, the early onset inherited forms are dominantly inherited. So if your dad or mom had the gene mutation, you're going to get the mutation and you have a 100% chance of developing the disease early in life. And there, I always thought in that case, geez, if I was like if I my parent, I knew they had the mutation, and say I was whatever 18 years old, thinking about getting married, having kids, that I would first get genetically screened, see if I have the mutation, and then if I did, and my wife and I still want to have kids, then there are ways you can actually do in vitro fertilization and embryo selection, and actually, so you could, in theory, you can actually make sure that your child does not have the mutation with currently available technology. But there's some ethical considerations there. And I guess it's not going to work so well for these childhood disorders because, well, how many, so in some of these disorders, do the individuals reach reproductive age and have kids, or it depends, I guess, on.
It does depend on the form and the phenotype. So there are some forms of NCL, most forms of NCL, they start very early in childhood, and lifespan is can be very short. Short as in age 10, age 13, sometimes still short, early 20s, 30s. For those who have a lifespan that extends into the 20s and 30s, by those ages, they're usually fully dependent for all of their care needs. And so that's reproduction is not typically something that we see. There are forms that are very attenuated, where vision onset loss may actually start in adulthood, or there are forms that are allelic with spinocerebellar ataxia, so onset might be in the 60s. Those are even among the rare, those are very, very rare forms, or at least ones that we don't recognize as often. For the childhood onset forms and the opportunity for parents to consider whether or not they would like to pursue pre-implantation genetic diagnosis, that too may depend on the form. And what I mean by that is there are some NCLs, like the classic form of CLN1 disease or classic form of CLN2 disease, where the onset is incredibly early. Infantile NCL may start anywhere between 6 months of age and, say 12 months of age. CLN2 disease might start between 2 and 4 years. CLN3 disease starts in elementary school years. You're thinking 4, 5, 6, 7, 8 in terms of onset. So the family that has a newborn and then in its infancy has this new diagnosis, they may not have actually finished their family reproduction yet, and they can take that information and think about it in their reproductive planning.
Yeah.
The family who has several children and then their, say, 7-year-old now has new onset of vision problems, may take a couple of years to get to diagnosis. They may have actually finished all their planned reproduction before they have the information. So we do see those differences in families in terms of number of children, whether they're able to pursue some of the reproduction advances in technology or not.
Okay, so you're in charge of the clinical trials. How many so you've been talking about rare genetic disorders, but Kennedy Krieger does, I mentioned traumatic brain injury. And by the way, my son's friend goes in, we go in like every weekend for 2 months. He was in a coma for several weeks. Then he came out. Then, over the next few weeks, he'd start to have some body movements. And then finally, after a couple months, he'd like start to speak a little bit, slur his speech. But anyway, he ended up getting so much better. He went to high school, graduated high school, even went to college. He still has some movement problems. I guess there's some, permanent problem in the motor system, a little bit of speech problem, but otherwise he's cognitively, so he had a really good outcomes. So you're overseeing the clinical trials. How many clinical trials are going on if you take like everything, or do you know? I mean, there may be so many. But...
Well, first I want to say I'm really glad to know that your son's friend had it sounds like a long course, but a positive course and a positive outcome at the end, even with residual kinds of symptoms that he's managing. It doesn't always happen that way.
Right.
And so I'm glad that he was able to get the care and support that he needed, and not too far from home as well. That's often a big issue, is that there might be the care available, but it could be across the country or 10 hours away. So I'm glad that he was able to have that right within the region. That's hugely important. At Kennedy Krieger, in terms of our clinical trials, around the world, there are thousands of clinical trials happening at any one time. And as we build our focus on clinical trials, we have more than 50 trials that are running at the center right now, and they bridge a variety of diseases. Fragile X syndrome. We completed a series of studies on Rett syndrome, and we were really delighted to have the opportunity to support studies that led to FDA approval for the first therapeutic treatment for Rett syndrome. Connie Smith-Hicks led those studies at Kennedy Krieger. We have work in osteogenesis imperfecta, in epilepsy, in anxiety. So really a wide variety of conditions, including cerebral palsy, neonatal stroke, and others. And so our group, our clinical trials unit, the goal of our team, we have a team of about 20 professionals focused on clinical trials. We want to make it easier for people to participate in clinical trials and for faculty to lead clinical trials, whether they have an idea that they'd like to pursue and are looking for support in design and development of that study, or whether they actually have deep clinical expertise in an area and they're approached by a company to establish a site. Our services really serve to provide strong operational support for those studies, and then on the scientific design side of things to really think about how we develop and design rigorous clinical trials. So it's a great resource, and it has been really exciting to grow and build.
The NIH has a kind of a special, well, that NIH FDA, I guess, kind of together, special track for rare disorders where clinical trials can be approved a little easier than with some more common adult disorders, right? So there's this, in 1983, there was this Orphan Drug Act. Can you talk about orphan drugs and kind of the accelerated pathway to developing treatments?
So in 1983, if you look at the time before that, there really wasn't much interest in rare disease drug development on the part of pharmaceutical companies. Certainly the people impacted by those diseases have always been interested in development, but it was hard to establish partners. Maybe there was one new drug approved, maybe two each year, some years with nothing approved. And so the goal of this act was to actually spark interest by pharma. And one of the main barriers was the perception that there really wasn't a tractable financial path to develop drugs for very small populations of people. And so the Orphan Drug Act first created the US-based definition for rare disease. So when we talk about rare diseases in the United States, we mean a condition that affects fewer than 1 in 200,000 people in the United States. That definition comes directly from the Orphan Drug Act. And the other piece that it established, the important piece that it established, were financial incentives to companies who were pursuing development of treatments for a rare disease that met that definition. And those involve tax credits and waiver of certain user fees. And it's a financially driven model. It has been very successful. So almost right away, you start to see this increased interest. And when you look over the past decades since the initiation of the Orphan Drug Act, we've gone from 1 or 2 a year where there are years where there are 40, 50, 60 new drugs approved for rare diseases specifically. So it's been wildly successful in that way. So the Orphan Drug Act itself is a financial incentive model. There are also programs that the FDA has that are really trying to accelerate the development of rare disease therapies by recognizing need and providing non-financial sort of incentives in terms of partnership, advice, guidance, shortened timelines by the FDA. And those fall under different sorts of pathways. There's the accelerated approval pathway. And that actually originally was really brought to the forefront in the HIV era. And the accelerated approval pathway relies on the use of a surrogate endpoint, meaning a biological or biochemical marker or measure that's reliably likely to predict clinical benefit. Clinical benefit meaning how someone feels, functions, or survives. This is a different kind of measure than that, and that's predictive of those kinds of endpoints. So for HIV, that might be HIV viral load, predicting transition to an AIDS diagnosis or an AIDS-defining illness or even lifespan. And so that has been used quite a lot in the oncology setting. And now we're seeing that used increasingly in neurological conditions, to some controversy. But I think biomarkers like dystrophin and Duchenne muscular dystrophy, the most recent approvals related to Alzheimer's disease that, again, have had some controversy.
Yeah.
Several of those have followed an accelerated approval kind of pathway. And they result in initial conditional approval, still go through all of the same rigor to transition to a full traditional clinical approval. There are other kinds of pathways that have to do with breakthrough therapies. If it's a condition that's never had a drug approved, is really in need of targeted therapies, there may be things like that too. So you have financial incentives, but then you also have these other pathways as well. There are several others.
Yeah. So a lot of activity. And are any of the trials going on at Kennedy Krieger involved? Well, yeah, we're involved in the enzyme replacement, but what about gene therapy?
So gene therapy has really exploded. I think there are advances that have made the human implementation of gene therapy trials more tractable after a period of quiescence. A lot of that has to do with vector delivery advances in the use of vectors that can target specific cell populations, identification of vectors that very well target the CNS, advances in understanding of optimal routes of administration for the CNS. So again, we see a lot of advance there, and that has translated into an increasing number of clinical trials. We're pursuing some of those trials at Kennedy Krieger. And I think that those trials, it would be wonderful to see more rapid translation of this potential into approved therapies. And I think some of the challenges have to do with, yes, the small populations, but also the readiness within those specific conditions to support rigorous trials. So while on the biological science side of things, or on the genetic side of things, you may be able to identify a target, develop a reasonable preclinical model, test that therapy. On the human side, it doesn't actually mean that we know in sufficient detail, when does the disease start? How does it unfold? How do I measure this and measure symptoms? And how would I know? How do I quantify success when it occurs? And those pieces, they seem like they should be obvious, but they actually really can take quite a lot of work. And that's where a lot of our focus in Batten disease is trying to be sure for that all 13 of those forms, when there are compounds ready for testing, we've thought about those issues. We've developed the relevant partners, and we can sequence symptoms, have ready measures, and be able to receive new therapies in a way that means we have trials with outcomes that are positive or negative and fewer that are inconclusive.
Yeah, there's a lot of technical hurdles so the human brain's a lot bigger than mice, and like if you use a adeno-associated virus A, B to target neurons, that's easily doable, but then how many neurons do you have to target, and how are you going to get all of them infected with the virus? And it's yeah.
That's the key question. I think that some of that issue is being considered in the vector, but also in route of delivery.
Yeah.
So while there still are, depending on the disease, depending on the nature of the intervention, you still may see targeted intraparenchymal delivery of investigational drug. But more often, I think we are seeing intrathecal, intracisterna magna, interventricular administration into the CSF for greater biodistribution, or intravenous administration or combined CSF IV approaches. So depending on the disease, there are still times where it is quite relevant to have a targeted intraparenchymal approach, but I think the scaling from mice to human, from mouse to human-sized brain, has had those challenges that you end up with this very small radius around the area where the drug was delivered and really not sufficient distribution with intraparenchymal targeting. But it depends on the need, because that's not universally true. Sometimes there is a very focused and targeted need, in which case you want to target that brain region and not the whole brain.
I myself actually have a rare genetic disorder, but it's unusual in its late onset. It's primary erythromelalgia, and it's it was kind of strange. I've had a lot of pain issues my whole life, but it's gotten worse, and I got this neuropathy now in my feet, and my feet turn red, and it's caused by a mutation in a sodium channel in the nociceptive neurons, the neurons that convey pain. And actually, it was pretty interesting because I don't know if you've come across Matt Hoke.
Yes.
Yeah. So he's a colleague of mine. Actually, we helped him in some studies a long time ago, but he's my neurologist.
Nice.
And so he diagnosed me like 15 years ago with neuropathy, sensory neuropathy, but it was kind of strange. It's not diabetic. I got really good glucose regulation, and my conduction velocities were normal. And then they did skin biopsies and looked at the fibers, and the density was normal, but they were kind of tortuous. And so it was his idea for me to do genome-wide exome sequencing. So it turns out I have this mutation, but that specific mutation actually hadn't been described in the literature. So there's a guy at Yale, Steve Waxman, who's like the world's expert on these disorders. And I knew him, Matt knew him. So his lab actually made my mutation. So they have the DNA, cDNA encoding the NAV1.9, the sodium channel. They made my mutation and put it in dorsal root ganglion neurons and recorded from them. And they fire more easily. So I've been wrestling with that. The best thing for me to control it is exercise.
So you've just, you've described the journey for so many people with rare diseases. And, you know, I'm glad that you found something that helps, because it can be such a painful condition, that I hope that helps in that
The only
direction.
drug that really helps is actually duloxetine, which is used for anxiety. You mentioned anxiety and depression. That helps, but otherwise it's exercise. If I had some injuries a while back, couldn't exercise, and that was like my pain level went up and my mood went down.
We so often talk about exercise for prevention. And I think even for very specific diseases, we're increasingly understanding the role of exercise and the need for exercise, and actually the management of day-to-day symptoms. But I would say your journey, you know, one, you had a fantastic neurologist. I've had the chance to get to know Ahmet Hoke through our work together in the Annals of Neurology families of journals.
Oh yeah.
And so I do get I get a chance to hear his thinking and how he approaches problems, which is wonderful. But that's, you know, we start with what we think are common symptoms that might be
Right,
happening with
right.
someone, and we go to the most common things, but it does take someone to say, well, wait, does this actually fit exactly how I'm used to seeing this more commonly?
Right.
And that either push from the patient or family to say, let's keep looking, or from the clinician to say, I think there's more here, to get to that next step. And often we end up at an answer that might have some uncertainty, where, like you described, well, it hadn't been described before. And so then you have to think about, well, I was looking for something and I found something, but does that actually explain what's going on with the person who's here in front of me? And sometimes you can actually find the world expert and get them to sort of build a model, whether that's a cellular model or an animal-based model, or increasingly computational models to actually answer that question or to yield some insight. But that takes a lot of resources, and not every
Yeah,
person is
yeah.
able to
No,
sort of get to someone who can get that final answer.
no.
And it would be, I think there are a lot of people who are trying to work to make that equitable, available, because everyone deserves to understand what's happening with them, and then to be able
Yeah,
to take that next step to find targeted
yeah.
treatments.
Okay, Erika, I won't take any more of your time. I appreciate you taking the time. You're very busy now. I'm kind of semi-retired, and our granddaughter's here. She's a year and a half. She's a year and a half old, and I'm gonna go down and play with her now.
Well, that sounds great. I thank you for your time, and thanks for the invitation. I really enjoyed our conversation.
Okay, bye.
Bye.